Good morning, students. Welcome back to Pharmacology Made Simple. In today's class, we will be discussing about a very important topic that is called pharmacokinetics. So, what is pharmacokinetics? Pharmacokinetics is the study of the absorption, distribution, metabolism, and excretion of the drugs. That is, the movement of drugs into, within, and out of the body. Once the drug is administered, it is absorbed, that is, it enters the blood. It is distributed to different parts of the passage of the drug molecules across various barriers like the intestinal epithelium, cell membrane, renal filtering membrane, capillary barrier, and so on. Okay, so what is pharmacokinetics? It is the study of the absorption, distribution, metabolism, and excretion of drugs within and out of the body. So here you can see the image of pharmacokinetics. So once the person takes the drug, so the drug will be liberated, that is, it will be coming out from its membrane into the body, and therefore then it will get absorbed, it will get metabolized, then distributed and excreted. What are the mechanisms of transport of drugs across the biological membranes? There are two types of transfers. One is called the passive transfer and the other is called the carrier mediated transport. The passive transfer includes simple diffusion and filtration, whereas the carrier-mediated transport includes the active transport, facilitated diffusion, and endocytosis. Coming to passive transfer, first we have the simple diffusion. It is the transfer of a drug across the membrane in the direction of its concentration gradient. Remember this term, student? It is the diffusion which is happening in the direction of the concentration gradient. Lipid-soluble drugs are transformed or transported across membranes by simple diffusion after dissolving in the lipids of the cell membrane. Whereas in filtration, it is the passage of drugs through aqueous pores in the membrane. Water-soluble drugs with molecular size smaller than the diameter of the pores cross the biological membranes by filtration. Okay. So in simple diffusion, we saw that it is lipid-soluble drugs, whereas in filtration, it is the water-soluble drugs which are passing through the biological membranes. Here is an image where you can see the difference between what is a simple diffusion versus a facilitated diffusion. So in simple diffusion, what you are seeing is the diffusing particles are going with the gradient concentration and moving across the plasma membrane. Whereas in facilitated diffusion, you need a carrier protein in the membrane which will help in the crossing of the particles from one side to the other. In carrier mediated transport, we have the active transport. It is the transfer of drugs against the concentration gradient and it needs the energy. It is carried by a specific carrier protein. Only drugs related to natural metabolites are transported by this process. Example, levodopa, iron, amino acids. The compound binds to a specific carrier on one side of the membrane and moves across the cell. At the other side of the cell, the complex dissociates and the carrier moves back to transport another molecule. Okay, so you'll understand this better through this image. So here you are seeing in the active transport what is happening. There is something called the carrier protein, which is taking these drugs and it is moving to the membrane, and at the membrane side it dissociates. That means it opens up and releases those compounds to the other side, and this requires some energy. Next, we see the facilitated diffusion. It is a unique form of carrier transport which differs from active transport in that it is not energy dependent and the movement occurs in the direction of the concentration gradient. So, in active transport, we saw that it was energy dependent, whereas in facilitated diffusion, we are seeing that it is not energy dependent. The carrier facilitates diffusion and is highly specific for the substance. Example, uptake of glucose by cells, vitamin B12 from the intestines. Next topic is endocytosis. Endocytosis is the process where small droplets are engulfed by the cell. Some proteins are taken up by this process, like pinocytosis in amoeba. This process is currently being tried for delivery of some anti-cancer drugs to the tissues. So it is small droplets which will be engulfed by the cell, that means swallowed by the cell. Whereas the reverse process, exocytosis, is responsible for the secretion of many substances from cells. Example, neurotransmitters stored in the nerve endings. That is, release of the substances from the cells is known as exocytosis. So here we can see an image first of the facilitated transport where a soluble particle enters the channel of a membrane which is called the protein carrier. Then it gets bind to the receptor sites on the carrier and the carrier changes the conformation and slowly the carrier releases the solute on the other side of the membrane. So that is about the facilitated diffusion and it is not energy dependent. And here we have the image of endocytosis and exocytosis. In endocytosis, phagocytosis occurs. That means the engulfing of the particle by the cell is called the phagocytosis. And then after engulfing, it passes through the membrane. Here we can also see about the pinocytosis, how it is happening. The cell, it will engulf the particle, it will form a vesicle, that is a membrane around it, and then it moves into the cytoplasm. Whereas in receptor mediated endocytosis, there will be the specific receptor sites on the cell which will take in the drug. For example, the stars are the drug, and you can see the star is getting attached to the receptor, and then there will be a coated protein around it, and a vesicle is formed around it, and then it will get released. Whereas in exocytosis, we said already the cell itself is producing the drug, for example, the neurotransmitters. The vesicle or the membrane moves to the upper part of the barrier and then it releases the proteins. That is called exocytosis. Understood, students? Next, we see about absorption. Absorption is defined as the passage of the drug from the site of administration into the circulation. There are various factors which affect the circulation or the absorption of the drugs, such as disintegration, particle size, the area of the drug, the metabolism, the vascularity, the dissolution, the formulation, lipid solubility, pH and ionization, presence of other drugs, gastrointestinal motility, presence of food and diseases. Remember student, this is a very important topic. What are the factors affecting the absorption of the drug? Now we will see one by one. Several factors influence the rate and extent of absorption of a drug. The first one is disintegration and dissolution time. The drug taken orally should break up into individual particles, disintegrate to be absorbed. It then has to dissolve in the gastrointestinal fluids. In case of the drugs given subcutaneously or intramuscularly, the drug molecules have to dissolve in the tissue fluids. Okay, so if it is taken orally, it has to get disintegrated in the gastrointestinal fluids, and if it is taken the subcutaneously or intramuscularly, it has to dissolve in the tissue fluids. The second one is formulation. Pharmaceutical preparations are formulated to produce desired absorption. Inert substances are used with drugs as diluents like starch and lactose. Okay, so how it is prepared is also important for the absorption. The next point is particle size. Small particle size is important for better absorption of the drugs. Drugs like corticosteroids, grisofuelin, digoxin, aspirin, and tolubutamide are better absorbed when given as small particles. On the other hand, when a drug has to act on the gut and its absorption is not desired, then the particle size should be kept large. Example, antihelminthics like belfium hydroxynaphtholate. Okay, so generally when the particle size is small, there will be the better absorption. Next is lipid solubility. Lipid soluble drugs are absorbed faster and better by dissolving in the phospholipids of the cell membrane. So compared to water solubility, lipid soluble drugs are better absorbed and faster absorbed because of the phospholipids which are already present in the cell membrane. The next point is pH and ionization. Ionized drugs are poorly absorbed while unionized drugs are lipid soluble and are well absorbed. But the degree of ionization depends on the pH. Thus, acidic drugs remain unionized in the acidic medium of the stomach and are rapidly absorbed from the stomach. Example, aspirin and barbiturates. Basic drugs are unionized when they reach the alkaline medium of the intestine from where they are rapidly absorbed. Example, pethidine, ephedrine. Strong acids and bases are highly ionized and therefore poorly absorbed. Example, heparin and streptomycin. Okay, so here both you can see that the ionized drugs are poorly absorbed, as well as when the pH of the drug is more, such as the strong acids or bases, that time there will be the poorly absorption of the drugs. Next, area and vascularity of the absorbing surface. The larger the area of the absorbing surface and more the vascularity, that is more the blood supply, it will be better for the absorption. Thus, most drugs are absorbed from the small intestine because you know that the small intestine is a big organ and the surface area of the intestine is more and there is very good blood supply. So most of the drugs are absorbed from the small intestine. Next is gastrointestinal motility. Here we can see about the gastric emptying time and the intestinal motility. So in gastric emptying time, if the gastric emptying is faster, the passage of the drug to the intestines is quicker and hence absorption is faster. And in the intestinal mobility, when highly increased as in diarrhea, drug absorption is reduced. Okay, so if gastric emptying is faster, then the absorption is faster, whereas if the intestinal mobility is faster, the absorption is less. Next, coming to the presence of drugs. The presence of food delays the gastric emptying and dilutes the drug and delays the absorption. Drugs may form complexes with food constituents, and such complexes are poorly absorbed. Example, tetracyclines chelate calcium present in the food, because of which their bioavailability is decreased. So here, that is why when the drugs are uh, given to us, what the doctor tells us generally, either take it half an hour before the food or half an hour after the food. Why? Because the presence of food will delay the gastric emptying and it will also dilute the drug. Next, metabolism. Some drugs may be degraded in the GI tract, example, nitroglycerin and insulin. Such drugs should be given in higher doses or by alternative routes. And finally, diseases of the gut like malabsorption and achlorhydria result in reduced absorption of the drugs. So we have seen the 10 things which affect the absorption of the drug. This is very, very important. Next, we move on to something called first pass metabolism. It is the metabolism of a drug during its first passage through the gut wall and liver. It will help to reduce the bioavailability. The extent of metabolism depends on the drug and the individuals. Sometimes the dose will have to be increased for some drugs like propranolol. Propranolol is one of the drugs used for hypertension. Route has to be changed for some others like hydrocortisone. Okay, some of the drugs which undergo the first pass metabolism are morphine, chloropromazine, nitroglycerin, virampil, testosterone, insulin, and lidocaine. Okay, so what is first pass metabolism? Is this the first metabolism of the drug which occurs at the first passage to the gut wall and the liver. Okay, so here is an image of the first pass metabolism. So when the drug comes into the liver, you can see that 90% of oral medication is metabolized and destroyed by the liver before it goes into the circulation. Okay, and at the portal circulation, all the blood from the intestines is taken to the liver for the detoxification. And in case of oral medications, it will be in the stomach for at least 30 to 45 minutes. Whereas in nasal, the drugs are absorbed directly into the veins. Okay, so this is about the first pass metabolism. So in today's class, what are the things we have seen, students? We have seen about the pharmacokinetics, what are the different carrier methods or the transport mediums. Then we have seen about absorption and the factors affecting the absorption, and we have seen about the first pass metabolism. So what are the important questions which can occur from this topic is, first one, write in detail regarding the factors affecting the drug absorption. That can be asked as a 10 marks question. Then drug delivery transport methods for 5 marks, where you have to tell about the passive methods and the carrier mediated methods. Then we have the first pass metabolism, which can be asked for 5 marks. And what is pharmacokinetics for 2 marks? If you like the video, please subscribe and like the video. Thank you.